Hi everyone, tonight's video is on the process of translation. So you can see in front of you is the central dogma, and that states that the code in DNA codes for an RNA molecule, and the code in RNA codes for a protein molecule. We are going to be covering this last transition in the central dogma, and that is called translation. So RNA has to be translated into proteins because nucleic acid and proteins are in different languages. RNA is written in the nucleotides C, G, A, and U. Proteins are written in amino acids, and there are 20 different amino acids. How are these nucleotides translated into amino acids? What is the code? Could the code be in single nucleotides? In other words, could each individual nucleotide code for one amino acid? Well, that's only going to give us 4 different possibilities for amino acids, and that's not enough. We know that there are 20 different amino acids. Could the code be in doublets of nucleotides? Well, that would only give us 16 different combinations for 2 different places for 4 different nucleotides. And again, that's not enough. We need to be able to code for 20 different amino acids. Could the code be in triplets of nucleotides? This would give us 64 different combinations, and that's definitely enough. And it turns out the genetic code is written in triplets of nucleotides. Each triplet is called a codon, and one codon, which is composed of 3 nucleotides, codes for an amino acid. Scientists have actually deciphered this genetic code, and they know what codon codes for each amino acid. You do not need to memorize this codon table, but you will need to know how to use this codon table. So let's go through some of the unique things about this table. First of all, there's more than one codon for most amino acids, and that's called redundancy. If you look here, for the amino acid leucine, there are 6 different triplets or 6 different codons that code for the amino acid leucine. If we look here for the amino acid arginine, there are 4 different codons that code for arginine. And the same for glycine. There are 4 different codons that code for glycine. Now, the codons that code for the same amino acid but with 4 different codons, they tend to vary in the last nucleotide in the triplet. The first 2 are generally the same for each of the codons that code for these amino acids. So in the case of glycine, each of the codons that codes for glycine starts with a GG, but the 3rd nucleotide is the one that tends to vary. Scientists call this the wobble hypothesis. There are a couple of other codons that's very important for you to understand. And the first is that there is one codon for start. This is what tells the ribosome to start reading the the codons and the amino acids. And the start codon is always, for every protein, AUG. And this codes for the amino acid methionine. There are 3 different codons that tell the ribosome to stop reading the mRNA, and that's UAA, UAG, and UGA. So how does the ribosome translate the nucleotide code in RNA into the order of amino acids in a protein, as pictured here? Well, actually, it's not the ribosome that does the translating at all. It's actually another molecule called a tRNA. There are 3 players in the process of translation. There's the mRNA, the ribosome, and the tRNA, or transfer RNA. The mRNA has the code from the gene in DNA. The ribosome is kind of like the machine. It's the machine that assembles the amino acids into the growing protein. It actually forms the peptide bond between the amino acids. The tRNA is the translator molecule that speaks both nucleotide and amino acid. mRNA is made by the RNA polymerase in the process of transcription, as shown here, where it's going to read the template strand of the DNA, and using the base pairing rules, it's going to put in an RNA nucleotide into the growing RNA strand in a 5' to 3' direction. The ribosome is the machine, and it's made of ribosomal RNA and proteins. Ribosomes have 2 subunits, a large subunit and a small subunit. Then there's the tRNA molecule. tRNA molecules have this very characteristic and unique shape. RNA molecules are all single-stranded, But tRNA molecules fold up upon themselves and actually hydrogen bond within the molecule to form this very unique and characteristic shape. At this end of the tRNA, there are three special nucleotides called the anticodon. The anticodon base pairs with the codon in mRNA. Now, this tRNA with an anticodon will always carry an amino acid on its other end, and it will be the correct amino acid for the codon that this anticodon binds to. So how does tRNA translate the code? Well, let's look at our codon table here again. I'm going to look at this one codon in an mRNA, UGU. UGU would code for the amino acid serine. So a tRNA molecule could come in that has the anticodon AGA. This would base pair with the codon. The tRNA with the anticodon AGA will always be linked to the amino acid serine. So you can think of tRNA as speaking nucleotide on this end and amino acid on this end. And in this way, the code in the mRNA can be translated into the amino acids. There are 61 different tRNAs, and each has an anticodon that is specific to the amino acid it is carrying. To see how the tRNA, mRNA, and the ribosome work together to translate the code in mRNA into a protein, you should put the following link into your browser and watch that video while pausing this video. Once you have watched that video and you understand how these all work together, come back to this video to finish the final slide. So you should know, after watching the embedded video, what is a codon? Why are there 3 nucleotides in each codon? You should know how to read a codon table. Again, you do not have to memorize the codon table. You should know what are the 3 players in translation, and you should know their composition and their role in the process. You should know what is an anticodon, and you should know what direction the ribosome moves along the mRNA. So that's all for tonight.